Okay, welcome everybody to Monday Lunch Live. We've got a special Monday Lunch Live today. My name's Grant McCarthy. I'm the Executive Director of the Victorian Comprehensive Cancer Centre and also a clinician scientist at the Peter Mac. I'd like to begin by acknowledging the traditional custodians of the lands on which myself and all of today's audience are located, and pay my respect to elders past, present and emerging. And I'd like to extend their respect to other Indigenous Australians that are joining us online today. So we have some interesting speakers coming up in Monday Lunch Live, as shown here on this slide, and very much look forward to you joining us for those events as well. If I could have the next slide, thanks Cynthia. So for interacting today, you have three choices. You can use the Q&A function at the bottom of the Zoom webinar. You can raise your hand in participants for an audio question, or use the chat function, and we'll be monitoring all of those. So feel free to use whichever you would like, and you can ask questions as the presentation is underway, so we can manage all the questions in question time at the end. So today it's actually an absolute pleasure for me to welcome one of my colleagues in the melanoma field from the University of Texas MD Anderson Cancer Center, Dr. Jennifer McQuade. So Jennifer is an assistant professor of medical oncology and a physician scientist at the University of Texas MD Anderson Cancer Center in Houston, and has stayed up late tonight away from the family to be with us. So thank you very much for joining us, Jen. Jen's research focuses on how modifiable host factors such as diet and obesity impact response to treatment, melanoma biology and anti-tumor immunity. You know, in introduction, if I think there's any question we routinely get asked as medical oncologists when we first see a patient embarking on new cancer therapy, it's what can I do? What modifiable lifestyle factors in my diet, in particular, can I undertake to help with my treatment? So I know Jen today won't have all the answers for us, but she'll give us a wonderful exposition of some of the science that's rapidly emerging as to how diet and other factors can influence response to treatment. So Jen, really looking forward to your seminar, and over to you. Thank you.
Great. Thank you so much, Grant, for having me and for everybody for joining in. So I'm going to try to share my slides. We'll see if this works again. Hang on 1 second. Okay, am I feedback up?
All good.
All good, perfect. Okay, so yeah, thank you, Grant, for the kind introduction. And you're absolutely right. This is, you know, the most common question that we probably get in new consultations is, especially when we talk about immunotherapy and we're talking about, you know, kind of a 50-50 response in melanoma. The question always becomes, is there something that I can be doing to end up in that good response bucket? And particularly with diet and with lifestyle factors in general, a lot of the science behind this previously has been a little bit soft, and there's tremendous amounts of research in the field of prevention, but as far as how these modifiable factors such as diet, exercise, stress, etc., might actually impact treatment outcomes, and specifically cancer biology and anti-tumor immunity, really hasn't been very well investigated to date. So I'm really excited to share you some of our really early work on these factors. So just my disclosures. So obviously, immune checkpoint inhibitors have been really revolutionary in the treatment of multiple malignancies, and probably no place more so than in melanoma. So the prognosis for metastatic melanoma used to be easily described as just completely dismal, where less than 10% of our patients actually had survival greater than 10 years. And with the advent of targeted and then also immune checkpoint inhibitors, now we're routinely seeing patients that are living beyond 5 years and even potentially cured, to where we're basically taking patients with widely metastatic disease, eradicating their tumors and getting them off treatment and now observing them. And so now our 5-year survival rate is more on the order of 50%. But while we've spent a lot of times really celebrating these successes and these great tumor responses, we have to acknowledge that about 50% of our patients, even in melanoma, which is really our most immunogenic and most responsive to immunotherapy cancer, about 50% of patients actually derive no benefit whatsoever from these immune checkpoint inhibitors. And so there's been quite a lot of work, obviously, done on understanding what are the factors that determine response or resistance to immunotherapy. And traditionally, we think about either tumor intrinsic factors, where we look at things like oncogenic signaling pathways, so PI3 kinase pathway activations, Wnt-beta-catenin pathway activation, or tumor extrinsic. And here we really tend to mean the immune microenvironment. So is a tumor inflamed, or is there a lot of T-cell infiltrate in it? But one dimension that really has not been well studied to date is what about the role of host factors on immunity, and specifically on anti-tumor immunity. And so there have been more recent studies looking at the impact of age and sex, sometimes actually with directionality that's a bit contradictory to what we might think. So, for example, older patients, we would traditionally think that they would have impaired T-cell responses, adaptive immunity, and we know this from, for example, studies of viruses and vaccines. But actually, there's some indication that they do just as well as younger patients, and in some cases may actually do better. Now, these are host factors that patients can't change. These don't answer the question of, doc, what else can I be doing? But what about, are there other factors that may actually shape immune response? And so these include obesity, and I'll touch on that a little bit at the end of the talk. And then what patients are asking, what about diet, exercise, stress, sleep? And then what's become really fascinating is kind of a way to integrate all of these. So we know that some of these modifiable factors influence immunity in other contexts, again, from studies of viruses and vaccines and autoimmune diseases. But what's been very exciting is the role of the microbiome as really a prime target for enhancing immune response to cancer, and the fact that the microbiome seems to be a really strong integrator of all of these different host factors. So the microbiome within immunotherapy, this first came into context in 2015. There were 2 science papers that were published back-to-back, one from Tom Gajewski's lab at University of Chicago, and one from Laurent Zipfogel's lab in Paris. And basically what these studies showed was that in mice, that the gut microbiome could influence response to immunotherapy. And the way that these studies came about was actually pretty interesting. So we know that typically when we do mouse studies, it does not matter where I get my mice from. So in the U.S., we would typically get our mice from either Jackson Labs or Taconic Labs. And for melanoma studies, I will order female P57 black 6 mice, so the exact same genetic background. And now if I give the mouse a tumor and I treat them with chemotherapy, it does not matter where I got that mouse from. It could be from Jackson or Taconic. They have the same genetic background. I'm giving them the same tumor. I'm treating them the same way. They will have the same response. But what they observed actually was that the mice from Jackson Labs were responding to immunotherapy when they were injected with melanoma. And the mice from Taconic lab were not responding to immunotherapy. So again, same genetic background. The question became, what is the difference between those mice if they have the same genetic background? And interestingly, the mice, of course, they grow up in this, very specific environment. And it turns out that when you grow up in that specific environment, then you end up with a different gut microbiota. And all of the mice from Jackson will have a very similar gut microbiome from that environment. They're eating the same cow, they live in the same environment. Chronic mice have a very different gut microbiota. So they profile, they see these different gut microbiota. And then what they did actually was quite elegant. They did a fecal microbiota transplant from the Jackson mice that were responding to immunotherapy and the Taconic mice that were not responding. And now when you do that fecal microbiota transplant, then give the mouse the cancer and then give the mouse immunotherapy, you're actually transmitting responsiveness to immunotherapy. So the microbiota was not just associated with response to immunotherapy, but shifting the gut microbiome also actually shifted response to immunotherapy. So again, in two different labs simultaneously, they see that different gut microbiota influence response to checkpoint inhibitors. And so the question obviously became, is this just a mouse phenomenon, or is this also happening in humans? And so, Jen Morgo, who's a surgeon scientist at MD Anderson, and then I have to say multiple other groups at the same time. So Tom Gajewski's group in Chicago, Lawrence Zipvogel's group in Paris, decided to start profiling human fecal specimens as patients were going on to immunotherapy. And so this is the MD Anderson data. And what we see is that when they profile the gut microbiota, patients who had a response to immunotherapy have a much higher diversity of their gut microbiota, meaning there are more different types of bacteria, diverse bacteria within the gut microbiota. And then of those with the high microbiome, high diversity of the gut microbiome, then we see higher progression-free survival. And then if we drill down and we look at specific taxa, then there are specific bacteria that are actually associated with response versus non-response to immunotherapy. And then when we take, and now we look at immune cell infiltrate. So again, not surprisingly, as has been seen in study after study, patients that have a response have higher T-cell infiltrates in their tumor. So what's important here is that these bacteria that were associated with response are also associated with a more cytolytic T-cell microenvironment. So again, this is correlation and not causation. So now we come back to the mouse model and very similar studies conducted by the two other groups that published simultaneously. And so here now we take a fecal specimen from a human that responded or did not respond to immunotherapy. And now we do a fecal microbiota transplant from the responding human or the non-responding human into the mouse, inject the mouse with melanoma, and then treat the mouse with immunotherapy. And what we see is that the mice who received the responder human gut microbiota, their tumors now respond to immunotherapy, whereas those that receive the non-responder, their tumors grow unchecked. So again, we can transmit responsiveness to immunotherapy via a fecal microbiota transplant. So as I mentioned, so there was this one study at MD Anderson, there's multiple studies at other institutions basically showing that there are these differential signatures between responders and non-responders to immunotherapy. And this is not melanoma specific. And so they see this in renal cell carcinoma and non-small cell lung cancer. What's interesting, though, is that the specific bacteria that have been identified actually do not necessarily line up So each of these cohorts, there have been distinct signatures between the responders and the non-responders, but everybody's identifying slightly different kind of pro-response bacteria. And there's been a lot of questions in literature about why this is happening. And so one possible explanation is that everybody is handling these bacteria and these fecal specimens slightly different as far as the collection protocols, as far as sequencing protocols, et cetera. Another potential explanation is that we know, again, that the gut microbiota is actually heavily influenced by our environment. And so how does, say, living in Chicago versus living in Texas versus living in Paris, do we have kind of different environmental native gut microbiota, and how does that shape it? So the last thing to acknowledge here is that all of these early studies were done by 16S sequencing, which is basically the smaller ribosomal subunit, as opposed to now a lot of the studies are moving towards whole genome sequencing, which has a much greater depth. And so again, we might actually be able to kind of drill down further with that. So again, coming back to this question that our patients ask us, so I tell them the story about, the gut microbiome, about the patients being different, about now how we can change it in mice. And the first question my patients want to ask me is, well, what does my microbiome look like and how do I get a good one? So we know one of the things that's really interesting about the gut microbiome is that it is inherently modifiable. So only less than 10% of our gut microbiome is actually genetically determined. So I will have more in common from my gut microbiome perspective with, say, a roommate that lives with me than my brother that lives, 1,500 miles away and has a very different life. So a number of modifiable factors can be determinants of the gut microbiome, but a couple of the most important are medications, so antibiotic and probiotic use, but also things, antidepressants, PPIs, metformin. These are all things that in large population studies have been shown to influence the gut microbiome. And then diet is the other major determinant. And so essentially these bugs that live in our colon help us digest food. They help us harvest the nutrients from food that is otherwise indigestible to us. And they're essentially eating what we eat. And so diet is a very key determinant of our gut microbiota. So this actually is how I became interested in this realm. So I was very interested in diet, and when these studies came out, and we were looking at what are these pro-response gut bacteria, these are actually gut bacteria that we know quite a bit about from these large population studies. So the bacteria, say Calibacterium ruminococcei, that were associated with response to immunotherapy are actually bacteria that help us derive nutrients from fiber-rich plant foods. So there have been multiple prior studies in healthy populations showing that people that are eating more fruits and vegetables and whole grains have an increased abundance of these of these microbes, because these microbes are then helping us digest these fibers. When they digest these fibers, they then synthesize these microbial metabolites, these short-chain fatty acids, which we'll come back to a little bit as far as the mechanism of action. Now, from the French and the Chicago groups, there are other specific bacteria, Akkermansia, Bacteroidetes, that also have very specific dietary associations. And so in this case, interestingly, the French pro-response bacteria was associated with wine and coffee and chocolate. So again, these phenolic compounds. So basically these kind of staples of what really might be a healthy diet. So coming back to fiber, So fiber is what is termed a prebiotic. And a prebiotic here means that this is basically a food source for the bacteria. This is opposed to a probiotic where you're actually ingesting the bacteria. And that can be in the form of either a pill or of yogurt, kimchi, sauerkraut. These things are actually all probiotic foods. The prebiotic basically means that we're feeding the microbiota and feeding putatively what are putatively the beneficial bacteria. And then postbiotics, this is a term that basically means now once the bacteria have digested, have helped metabolize that particular nutrient, now what do they put out? And in the case of fiber, what we produce is these short-chain fatty acids. And so the hypothesis, when we saw that these that the bacteria that were associated with response were actually fiber fermenting bacteria, was that patients that were eating more fruits, vegetables, whole grains would have an increased abundance of these fiber fermenting bacteria. These fiber fermenting bacteria now, when we intake the fiber, now produce short chain fatty acids. And these short chain fatty acids are actually the primary nutrient source for our gut enterocytes, for the cells that line our gut. When we have short chain fatty acids, they're feeding the enterocytes, and we actually form a very nice thick mucosal layer. And so we have, and I hate the term, but we have a less leaky gut. And this, again, and now we start to get into a little bit of a hand waving about as far as how this actually specifically influences immunity and certainly immune response to tumor. But what we see is that overall, this microbiota gut interaction seems to kind of shape a overall tone to our immune system. And interestingly, too, these short-chain fatty acids actually have been shown to have an epigenetic role in conversion of our activated CD8 T cells into memory CD8 cells. So again, basically now we're looking at kind of a diet microbiota immune axis. So to interrogate this hypothesis within our patients, we started actually asking them about their lifestyle. So as patients were going on to checkpoint inhibitors, we were collecting fecal microbiota specimens and then also collecting questionnaires as far as their diet, as far as probiotic use, and then other measures such as depression, stress, physical activity that we're just starting to look at now. And so then we classify these patients as either responders or non-responders to immunotherapy. And when we first actually started this questionnaire, we just did a very brief National Cancer Institute dietary screener questionnaire. And the reason we started with such a brief questionnaire was actually that nobody thought the diet would matter, and we didn't want to burden our patients with too many questions. More recently, based on some of the results that we've been seeing, we've gone to more intensive 24-hour dietary recalls, which are actually administered by a trained nutritional epidemiologist. So again, preliminary data, but basically what we see is that very similar to healthy populations are patients that are eating more fibers, fruits and vegetables, whole grains, have an increased abundance of these bacteria that are associated with response to immunotherapy. And now this is what's called a PCOA plot that's basically kind of giving us a snapshot overall of the community structure of the gut microbiota. And here what we see is that those patients that are consuming a lower fiber diet have a very distinct gut microbiota from those that have sufficient fiber intake. So that's all well and good, but of course the most important thing is that does this actually is this associated with response to immunotherapy? And sure enough, we see that our patients that are eating a sufficient fiber diet are actually almost 5 times more likely to respond to immunotherapy and have significantly improved progression-free survival as compared to those patients that are eating insufficient fiber. We also then examined the interaction between the gut microbiota profile and fiber intake. So the type 1 signature is basically means this kind of pro-response bacteria, so high amounts of Ruminococcus and Faecalibacterium. And the type 2 signature is those with an unfavorable microbiota from the previous studies. So now when we layer those two together, basically those patients that have both the good microbiota profile and are feeding it with a high fiber diet are the ones that are most likely to respond, whereas those that have a kind of poor microbiome and are consuming a low fiber diet are actually the least likely to respond. And this actually might have important implications for studies of fecal microbiota transplant. So just as we saw in mice, where we're now doing fecal microbiota transplants to improve response to immunotherapy, now there are multiple ongoing studies in humans with FMT. And one of the questions remains, so if you do an FMT from either a healthy donor or from a patient who actually responded to immunotherapy, and you put that new microbiota into a person that's about to start immunotherapy or previously has not responded to immunotherapy, is that transplant durable, right? And so this is actually kind of interesting. So do you need, if you're putting these nice fiber fermenting bacteria into these patients, you actually need to continue feeding it with fiber in order for it to sustain. And it's actually a question that really fecal microbiota transplants have been studied for much longer in the context of C. diff or in inflammatory bowel diseases, and it really actually has not been looked at. So again, correlation does not equal causation, and there's a really good chance that these people that are eating fruits and vegetables and whole grains are probably also doing a lot of other things that are right in their life. So these might also be the patients that are exercising. They might have higher socioeconomic status. There's lots of potential confounders. And again, we adjusted for those as much as possible within the retrospective cohort. So to really assess causation, now we collaborated with the lab of Giorgio Trinchieri at the NCI. He was doing studies actually in mice looking at the impact of fiber there. And it turns out that mouse chow is actually a beautiful source of fiber. And so the way that you do fiber studies in mice is to do fiber deprivation. So now you take a mouse either on standard high fiber diet, or you do a fiber deprived purified ingredient diet. And now you inject the mice with melanoma and then give the mice immunotherapy. And so what we see is that just changing fiber very rapidly within 3 to 7 days completely shifts the structure of the gut microbiome. And basically these mice who are on the high fiber diet, their tumors respond to immunotherapy. So this is a green line down at the bottom. These are the tumors of the high fiber diet immunotherapy treated mice. Whereas here, these mice that are on a low fiber diet, there is no difference whatsoever between the tumor growth and those treated with isotype control versus with immunotherapy. And so basically fiber deprivation is completely abrogating the response to immunotherapy. Now, here what they did is they basically flow sorted out CD45 positive leukocytes from the immunotherapy treated either high fiber diet or a low fiber diet fed mice. And what we see is that basically only those mice that were on the high fiber diet within their tumor, are they able to really mount an effective T-cell response, an effective immune response. So fiber and diet in general, it has to be acknowledged, actually can have, can have multiple potential kind of downstream effects that may or may not all be microbiota driven. So for example, again, you could have polyphenols, you could have estrogens, you can have all of these other derivatives just from the food itself and the fiber itself. So asking the question, is this microbiome mediated? They then did experiments where they used germ-free mice, so mice that do not have a microbiota, and then did the same experiment now where we inject melanoma cells and then treat them with immunotherapy. And in the germ-free mice, there's actually no difference in response to immunotherapy between the regular diet and the fiber-free diet. So this is implicating that it's actually microbiome mediated. So what I've showed you thus far is that habitual diet in humans is associated with response to immunotherapy, and that in mice, that changing diet can change the response to immunotherapy. So the question becomes, in humans, in our patients, these ones that are now starting immunotherapy, they want to know both, do I have a good microbiome or a bad microbiome? But what they really want to know is, well, if I have a bad one, could I change it? And so the question is, with a dietary intervention, with a diet change, can we change the microbiome, or are we kind of set? And so this is a very elegant study that was done by investigators at the University of Pittsburgh, where they basically took African Americans living in Pittsburgh that were eating a very prototypical Western diet, of, lots of protein, lots of fat, lots of unrefined carbohydrates. And then they took Northern Africans in sub-Saharan Africa that were eating a very agrarian, plant-based, high-fiber diet. And what they did is they swapped their diet. So for 2 weeks, the African Americans now ate this very kind of maize, corn-based agrarian diet, and the Northern Africans ate a diet they were probably both very excited about at the beginning, and then maybe after a while didn't feel so good. So basically, kind of hamburger and French fry type diet. And so what we see is that here, the Northern Africans, as they're consuming the Western diet, this is their production, their potential production for short-chain fatty acids, so bacteria that can produce short-chain fatty acids. And what we see is that as they're eating this Western diet, then their short-chain fatty acid production potential goes down, down, down. And then on the converse, the African Americans now eating a high-fiber diet, their short-chain fatty acid production potential goes up, up, up. And we see the exact opposite now when we look at bile acid conjugation. So bile acids are actually the byproduct of meat metabolism. So again, as we're eating a more meat-based diet, some bile acid conjugators go up, 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 and they come down as we're eating a more plant-based diet. So this is a really important proof of principle showing that actually in a very short time, we can actually rapidly and reproducibly shift the gut microbiota by changing diet. So that's a pretty radical shift in diet. What about things that are a bit more subtle that, again, might be sustainable? And so this is actually one of my colleagues at MD Anderson, Carrie Daniel McDougall. She did a study in colorectal cancer patient survivors, where, again, in colorectal cancer, there's been quite a bit of studies linking the gut microbiota actually to cancer progression and with some specific bacteria there. And so what she did is she took patients and all she did was add a cup of beans to their diet per day. And so beans are actually this beautiful fiber source, prebiotic, very rich in other bioactive compounds. And basically the idea here was that by doing nothing else other than adding what is equivalent to 15 grams of fiber per day, could she actually change their microbiota? And what she sees is yes. So this is a patient. So this is it's a crossover trial where basically now patients are randomized up front to either getting the intervention or staying on their regular diet, and then after 8 weeks, then they switch. So this is a patient who initially started with the high fiber diet. And if you'll just follow the red, which is the Calibacterium, which is one of these pro-response fiber fermenting bacteria. So now they're here, they have this equilibration period where we're watching them. And now they go on this ramp up period where she starts with half a cup of beans per day. And now we go up to a cup of beans per day. And you can see that there's this huge bloom of the Calibacterium. So lots of increase in this bacteria because now we're feeding it what it wants. However, equally important, what we see is that now when we switch over to the control diet and we're no longer feeding those bacteria, then the amount of the Calibacterium goes down, down, down. So to change the microbiota, we can change diet, but we have to be able to sustain the diet to make a change. The other really important thing from this study actually is the fact that that there's quite a bit of inter-individual variation. So we all have different bacteria in our gut. And we kind of have our own natural community. And so each of these patients, so the circles are without the bean intervention, the triangles are with the bean intervention, and you can see that they're all kind of within their own little stratosphere. So it's not like pre-intervention, everybody is over here, post-intervention, everybody is over here, clustered together. Instead, it's like you shift your own native gut microbiota a bit, but it's not like everybody has this one phenotype that's like the post-fiber and a one phenotype that's the pre-fiber. But when we now look, and so this is actually, this top is looking at the specific taxa that were present after the fiber intervention. And so we can see that, these colors, everything is shifting, right? So this is one patient looks like this. This other patient has tons of Calibacterium. So are we really doing anything, right? There's all this noise. There's all this change. Does that actually change anything? So now, if instead of looking just at the specific taxa, now we collapse this, and we're actually looking at function. What we see is that now we're collapsing this heterogeneity. So when I may have at baseline a higher amount of Calibacterium in my gut microbiota, my colleagues may have more ruminotoxins. They are both fiber fermenting bacteria. When I eat more fiber, I'm going to bloom the bacteria that was already present in my gut. My colleague is going to bloom the bacteria that was already present in her gut. It doesn't actually matter what the specific bacteria is. What matters is what it is doing. So these are now, again, fiber fermenting bacteria. There's lots of redundancy. What matters is that they're performing the same function, producing these short-chain fatty acids, and again, potentially, right, impacting immunity. So I've showed you so far that habitual diet is associated with response to immunotherapy and specific microbiota profiles, that in mice, that we can change diet and change response to immunotherapy, and that in humans, we can change the diet and change the microbiome. But the question still is that can we change diet and actually change response to immunotherapy? And so what we're doing is we're now conducting studies that are very similar to that study that was conducted at the University of Pittsburgh of what are called controlled feeding studies, where we actually provide patients with every calorie containing food and beverage that goes in their mouth over the duration of a study and longitudinally profiling their gut microbiota and systemic and anti-tumor immunity. And the reason we're doing that is because as motivated as our patients are, diet change is hard, which any of us that have ever tried to go on a diet and lose weight would know. And so by providing patients with all of the food, then we're taking out that behavioral component. Because what I want to know is that if I change the diet, does it change the microbiome? Versus if that fails, just like, for example, with a novel targeted therapy, was it that I didn't shift, it didn't change the target, or was it that the target wasn't important? So this way, by giving them all the food, I can know actually was the target important. If I really controlled and changed it, then can I change the microbiota? So we're studying this. We're basically trying to, really test diet as a precision intervention, just like we would with a drug. So in phase 1, this is the first time we're ever conducting this type of controlled feeding study at MD Anderson. So we're actually just testing the feasibility, the palatability of giving patients this food. Will they eat it? And we've actually, in our phase 1 study, have over 95% compliance with a dietary intervention. In phase 2, now we actually move to patients that are on active immunotherapy. And again, our longitudinal profiling, does the gut microbiome change? Does systemic and anti-tumor immunity change? And then the idea after these proof of principle studies was that we would then move on to phase 3 behavioral intervention studies. So now I've proven that in a controlled fashion, if I change the diet, I can change the microbiome, I can change immunity. Now, am I able to teach people how to change their diet, and in a larger scale and multi-institutional study with lots of patients, am I now powered to actually look at change in disease outcomes? So again, this is our phase 1 study that's actively recruiting. It's actually a 6-week intervention where we take people and we take them up from a less than 20 gram of fiber diet, which is the average fiber intake, to a 50 gram of fiber diet, and we ramp them up over this period of 6 weeks to see are they able to tolerate this, and then longitudinally profile the gut microbiome and then look at systemic immunity as well. The other part of this is that at 12 weeks, so they get the diet for 6 weeks, and then 6 weeks later, we ask them about their diet. And in the initial patients that have gone through, even though we have not told them to stay on a high fiber diet or have not continued to counsel them, all of our patients have actually maintained a higher fiber diet 6 weeks later. So it's really fun to actually conduct this type of research. So we have a trained dietitian chef that prepares all of the food, and then we could, of course, taste test it. And then the trial that's just now underway is actually our trial in patients on immunotherapy. So now this is a randomized control trial where we either take patients and they get a healthy American Institute of Cancer Research, American Cancer Society control diet that's, again, whole foods based. Still, the recommended amount of fruits and vegetables, low in red and processed meats, etc., but mimicking the average fiber intake in this population versus then a high fiber diet where they're ramped up to 50 grams of fiber per day. And now in this study, they're actually getting the diet for 10 weeks. The patients that have metastatic melanoma at baseline are actually getting pre-treatment and then post-treatment paired tumor biopsies. And the idea is that we'll do multidimensional immune profiling and then also do metabolic profiling of both the fecal specimens, the blood, and then the tumor as well to really understand how those all interact together. So again, how do we now translate this actually into a phase 3 setting? This is all just a controlled feeding study. The idea is that we now move into a behavioral diet study where we could really look at does this actually change progression-free survival and overall response rate. I will end actually just on this question that's always asked. Is there something easier that I could do to change my microbiome? Again, we know that diet is hard, so couldn't I just take a pill? And probably prompted by all of this really interesting work going on with the microbiome, we actually know that 42% of our patients in this cohort were actually taking an over-the-counter probiotic. And those patients taking a non-selective over-the-counter probiotic actually had a lower diversity of their gut microbiome and were actually less likely to respond to immunotherapy. And so, again, plenty of potential confounders here. Did they have some type of gut issue at the beginning, which made them want to take a probiotic? Could they have been taking antibiotics? We actually removed the antibiotic users from the cohort because we do know that that influences response to immunotherapy. We removed them, and those were actually the results from the without antibiotics. But there's also this study from an Israeli group where they took patients or mice and gave them broad-spectrum antibiotics, and they did one of three things. They either just kind of let them recover with time, which you actually recover your microbiota with time. They did a fecal specimen from before they started on immunotherapy, and then gave it back to them before they started on antibiotics, and then gave it back to them afterwards to an autologous fecal microbiota transplant, or they gave them probiotics. And what they saw is spontaneous recovery. Again, with time, you recover your gut microbiota. With the autologous FMT, you immediately recover it. And actually, the patients that received the non-selective probiotics that actually had the slowest recovery of their native gut microbiota. And when they took the filtrate from the fecal specimens from the mice that had the probiotics, what they actually see is that there's basically like this competitive exclusion. Where the lactobacillus seems to be actually secreting something that's keeping the other good bacteria from growing. And then just to touch on, again, can we simplify instead of doing a diet? Could we maybe just look at single fiber sources? So a lot of ongoing work in this area, looking to maybe just do a fiber supplement or prebiotic supplement. To actually influence the microbiome in response to immunotherapy. So this is some work out of Bernay's lab suggesting that maybe inulin could do this. So just a conclusion, the gut microbiome appears to be distinctive between responders and non-responders to immunotherapy, and that in mice, we can transmit responsiveness to immunotherapy via fecal microbiota transplant. And there are multiple clinical trials now testing this in humans. Diet and biotics, so either probiotics or antibiotics, are actually very key determinants of the gut microbiome and can harm or potentially improve response to immunotherapy. And then ultimately, really, diet is a precision intervention we're doing this controlled feeding study here, we're not doing baseline gut microbiome profiles. I'm making sure the patients are not, for example, eating a vegan, very high fiber diet at baseline. But the question, just like with fecal microbiota transplant, is actually how do we choose the right patients for this intervention? So, for example, can I just screen patients and say, hey, you're not eating enough fiber, we need to increase your fiber? Is it based on their baseline gut microbiota? And then is this the right diet for every patient? So this is in the context of immunotherapy. I did see a question come through in the chat, do we think that this would be translatable to other malignancies? Yes, within the context of other malignancies treated with immunotherapy, but what about interactions with different therapies, such as targeted therapies? There's been some really elegant work out of Lee Cantley's lab at Cornell showing that, that a ketogenic diet might actually synergize with PI3K inhibitors that cause hyperinsulinemia. And so if we're looking at diet as a precision intervention, is it based on the host metabolic phenotype? Is it based on the gut microbiome? Is it based on what therapy they're getting? Is it based on, kind of their key oncogenic signaling pathways or tumor metabolism? So a lot of interesting unanswered questions there. I'm just going to skip ahead to my acknowledgement slide. So all of this microbiome work was with the mentorship and the collaboration of Ben Wargo at MD Anderson. The Dietary lifestyle surveys were initially a project of a grad student in our lab, Chris Spencer, who's now at the Parker Institute, and then Daniel McQuade is the nutritional epidemiologist on all of our studies. And then outside the mouse work of the fiber that I showed you was all Giorgio Trinchieri and Marie Zedeshu's work. So happy to take any questions.
Great, thanks very much, Jen. Fascinating area, and I have to kick off with a comment. I think there's many biotechnology companies that if they saw effect sizes that large from their drugs, their stock prices would be rocketing. So it's really very interesting. So we've got some questions that have come through from the audience. Now, Lani Morrison asks a good question on chat about testing the blood. And do you think, testing blood might be a way of looking at the microbiome at all, Jen? What are your thoughts?
Yeah, so right now, all of the studies that have been done are all with fecal specimens, and again, we're looking for the specific bacteria. I think one of the things that will be interesting in looking at blood is looking at the metabolic output of the bacteria, and so again, how do we link kind of what's going on in the gut to what's going on systemically to then what's going on in the tumor?
Yeah, I was really taken with that fantastic slide you showed about the diversity in the 16S sequencing between individuals, but then when you looked at the microbiome metabolome, there was, a lot more, less heterogeneity, but nonetheless, there were some very specific, distinct differences between individuals. So do you think that offers some hope if you believe that, taking a pill is easier than changing diet, of identifying, some specific microbiome metabolic interventions that could be developed as drugs, or is that just too hard and we're best sticking with diet?
No. I don't think it's too hard. I think that we're so early in this science that I think that that's where it will go, to where again we can do hopefully more targeted microbiome interventions. Diet's interesting because there's lots of other mechanisms by which it could be beneficial. So it might not all be microbiota mediated, but exactly, maybe we can profile the microbiome look at specific taxa, look at its metabolic output, and then how do we specifically target that in an individual patient. So no different than what you're doing with, with targeted therapy for the tumor itself.
Yeah, great. So can I just follow up on the question about pills and easier taking pills in some people's views than changing behaviour? So the use of antibiotics. So do you think that antibiotics may be a therapeutic approach to change microbiota? Or are there potentially good antibiotics and bad antibiotics, and that you can sway a microbiome towards a more favourable microbiome by the use of antibiotics?
Yeah. So it's a good question, and there's a few different ways to think about it. So the first is that, so in mice, we know that one of the ways to shift the microbiome is to use antibiotics, and we can basically kind of use antibiotic ablation to reset the microbiome. Before fecal microbiota transplants are performed, then we also tend to wipe out the native gut microbiome with antibiotic ablation, but then you're giving them back something good. So yes, so there's, we've talked a lot about potential good bacteria. What we didn't really discuss is that, again, there's bad bacteria that could also be negatively correlated to response, and is there a way to wipe those out? In human populations, the data that we have thus far is, again, very good. So there's been many studies now showing that patients that were given an antibiotic within 30 days of initiation of immunotherapy have decreased responsiveness to immunotherapy. Again, though, lots of potential confounders. So those patients, right, were potentially sick. They were the ones that needed an antibiotic. We weren't just randomly prescribing antibiotics to one group versus the other. And so certainly confounders there. Some more nuanced work has been in the leukemia stem cell transplant population, where there's all of those patients get an antibiotic at some point, right? Once they're completely, their immune system is completely wiped out. There, what they find actually is that it depends on the antibiotic. So most of the bacteria that are beneficial that live in our gut microbiome are actually anaerobic bacteria. And so it tends to be these broad spectrum antibiotics with anaerobic coverage that are the ones that are associated with worse response. So again, that hasn't been studied in the context of checkpoint inhibitors, but I do think that will start to come out where certain antibiotics are better versus worse. And again, is the right approach to target increasing the good bacteria versus wiping out bad ones.
And Jen, there's a question on Q&A from Meredith Cummins about carcinoid syndrome. So there's a challenge if patients have disorders that are associated with diarrhea or GI tract abnormalities. Do you see challenges extending these findings into patients with GI disease?
Yeah, no, that's really interesting. And again, I don't know of any studies so far in these kind of more rare tumor and conducted. As far as the GI malignancies, so in general, again, there's been quite a lot of work just showing how the gut microbiome might actually influence cancer progression within colorectal cancer. And then some really cool work recently looking at pancreatic cancer and the gut microbiome and the tumor microbiome, where they're actually seeing differences in the tumor microbiome between patients that have long-term survival after pancreatic cancer versus those who don't. And then in mice, where the fecal microbiota is actually reversed transmigrating in pancreas and influencing the tumor microbiome. And now in the mice, you can do FMT and actually change not just the gut microbiome, but pancreatic tumor microbiome. And so now there's some trials of that in the perioperative setting in pancreatic cancer.
Thanks very much. Well, we're up to time, everyone. And I think in that last answer there, Jen, exploring in a future seminar the tumor microbiome would be very interesting as well. So, look, it just remains me to thank Jen so much for giving up your Sunday evening in Houston to join us for what was just a wonderful seminar. And Jen, I think I can see a time coming when in your disclosure slides, you'll be disclosing food companies and not just... Yeah. So thank you so much for a wonderful seminar, and everyone look forward to seeing you on forthcoming VCCC Monday Lunch Live. Thanks, everyone.
Awesome. Thank you. Okay, take care.